Okay. So we have a few things to do. We may not finish today. Whatever we don't do today, I won't put on your exam. Okay, obviously. So how far we get will dictate what's on exam 1. But sections 3.4 and 3.5, there's a lot of material in it. It goes over all the methods, and it's very introductory. A lot of those methods we're going to dig deeper in the lab. Okay, so really just understand the big picture of these methods. And then we're going to start getting into real cell biology here in the last couple sections when we're talking about non-coding sequences, which is going to be a lot more new material for you guys. Last time we didn't finish section 3.3. There was dideoxy sequencing that we need to go over. So, all right. So I actually want to make a little adjustment here. So I don't like how the book goes over dideoxy sequencing before going over PCR. So I'm going to just jump ahead and go over PCR and then come back to section 3.3, because you can't understand dideoxy sequencing without understanding PCR. Yeah. When is exam 1? Next week. Yeah, on Wednesday. So it'll be the first thing we do when we come in. Okay. Okay, so PCR stands for polymerase chain reaction. And it is a method of amplifying DNA. It amplifies DNA very specifically according to the primers that you use to designate where you're going to amplify the DNA. And the reason that's so useful is, when you, let's say, let's give an example of forensic science, right? So you get a DNA sample. Let's say you get just one red blood cell from a crime scene. There is not enough DNA in there. Like the amount of DNA that you're looking at is like picograms of DNA to do anything with, except detect it. That's really the only thing you can do. You can't sequence it. You can't clone it. You can't do anything with it except detect it. And PCR is a very, very sensitive technique that enables DNA detection, specific detection, using primers that are complementary to the sequence that you want to amplify. So let's say we get that red blood cell. So we've got a big chromosome and we have 0.005 nanograms of total DNA here. And we want to be able to clone a gene that's found in that chromosome. So you have to have sequence information to do PCR. If you have sequence information, you can generate primers that flank the region that you want to amplify. We call this an amplicon, which is the region that you're amplifying. All right, primers come in the forward and the reverse. And these oligonucleotides are about, anywhere from like 15 to 25 base pairs long, and they're complementary to the sequence in that flank the region that you want to amplify. Okay. Now, DNA replication, this like in vitro synthetic DNA replication was done for many years before 1988, but it was a real pain to do. Right? To do synthetic DNA replication, you basically have to do 3 things. First, you have to denature the DNA. And what that means is you have to expose the DNA to a particular temperature, which is typically 95 degrees Celsius. At 95 degrees Celsius, you break the hydrogen bonds and the 2 strands of DNA come apart. So now you've exposed the nucleotides to potential primers that you want to bind to it. The second step is getting those primers bound to the DNA, which is an annealing step. And that usually happens at about 65 degrees Celsius. All right, so we separated the DNA, and now these forward primers have bound to the complementary regions of the DNA. The last step is extension, which typically happens around 72 degrees Celsius. Right? And here you have, DNA polymerase that's in your reaction tube that binds to the primers. We'll learn later on, DNA polymerase cannot synthesize DNA de novo. It can't do it on its own. It needs a primer to initiate it from. And then during extension, it binds and it amplifies. So after one round of this, now we've basically doubled the amount of DNA. But if you have a way of doing this, let's say 30 rounds, which is a reasonable number of rounds that you do PCR, you exponentially increase this amount of DNA to an amount that you can actually visualize it on a gel, that you could clone it. You're now up in like the nanogram range of DNA. All right, so this was done for many, many years before Kirby Mullis came up with this brilliant idea, but it was a real pain because every time you did one of these reactions, you had to purify the DNA and you had to basically start all over. It would take a week to do this. So I keep calling him Kirby. His name's Kary Mullis. Kary Mullis is a real interesting figure. He got the Nobel Prize for this. And during the award ceremony, he went surfing. He didn't show up. But during the O.J. Simpson trial, he was actually brought in as a witness from the defense. And because of his prolific drug use in college, they were trying to, say that his stuff had no merit. But this is a very, very, this changed, revolutionized molecular biology because we have now ways of targeting specific sequences and working with the DNA. So what he came up with is using a DNA polymerase that is stable at these high temperatures at 95 degrees. So he went to Colorado and he noticed that, those springs out there, which are at a practically boiling temperature, have bacteria in it. And the bacterial strain that he isolated the DNA polymerase from was called Thermus aquaticus. So that bacteria can replicate and synthesize DNA at a very high temperature. So now in one reaction tube, you can add the primers, the DNA, the polymerase. And when I say DNA, I mean template DNA. And then also you have to obviously have in that reaction DNTPs, right? We learned about DNTPs. These are nucleotide triphosphates. They have the three phosphates, the A's, the T's, the C's, and the G's. And then you use a thermal cycler, which can cycle through these three temperatures over and over again. And we'll do PCR in our lab. Okay, so you can see this figure here. You've got you denature, you separate the strands of DNA, you bind the primers, all right? So, there are machines that will generate oligonucleotides rather quickly. And in your lab, when you're ordering these primers, you just do it on the computer, you punch in the code that you want, and they ship them to you next day. It's very standardized. But after you're exposing it to 72 degrees and you amplify this region, now you have so much DNA in that tube of this specific region, it like washes out, that template DNA. You can't even work with that. Okay, so that's PCR. A lot of the techniques that we do are kind of revolutionized by this first technique here, PCR. All right, so after PCR was invented, discovered, whatever. For what? It's a range. I wouldn't ask you what temperature it was, but I might indicate, maybe in the lab, it's a range of temperatures. So the annealing temperature actually is very sequence specific. So because it's related to the melting temperature, it's the temperature at which the DNA like completely unravels. It depends on the code. If you have a lot of GC content, it's going to be a higher temperature. If you have a lot of AT content, it's a lower temperature. So what did it say on the board? What I have? 50. That's a little low, but it's a range between like 52 and I'd say 68 is a range. Okay, so good question. So in order to understand basic dideoxy sequencing, you need to understand PCR because the first step in dideoxy sequencing is a modified version of PCR. All right, so I would encourage you to pay good attention to this. It's gonna be a little tricky to grasp the first time, but some of you may have talked about it in Bio 1. I think the best way to actually approach this is to look at it backwards. Right? So we'll talk about that modified version of PCR last. And first off, just look at how like a big DNA sequencer works. So this machine has in it a capillary tube, which is a really thin tube that contains a very pure form of agarose. So you remember agarose gels, right? Smaller fragments migrate faster than the larger fragments. So if we had a sequence here, it's a really short sequence, but dideoxy sequencing enables you to sequence DNA up to around 300 to 400 base pairs. All right, so what the modified form of PCR is going to get for us are fragments that are representative of this sequence. So we're going to get a fragment that's like this, and then just like this, this. Right, so the last nucleotide of each of these fragments you could assemble together, right, to generate this sequence, the original sequence. This fragment is smaller than this fragment. So if we had a pool of these fragments together and we pushed them through this capillary, this is going to come off first, this is going to come off second, and so on. Also, if we had a way to label this final nucleotide and give it some fluorescence, let's make the A's blue, the T's green, G's red, and the C's orange. If we had a laser that could shine on the piece of DNA when it comes off the capillary, and if it fluoresces whatever color, then the detector can detect the fluorescence and associate it with a particular nucleotide. Right? So that's what the actual DNA sequencer is doing. It's you're separating out all of those representative fragments, and then the computer is putting the fluorescent levels together, which are right here. It's called a chromatogram, and it's using that to generate the sequence. All right, so how do you get all of those representative fragments? Well, it's a PCR reaction that just uses one primer. So let's say this is our piece of DNA, our template DNA. We just put one primer where we want to start to read the sequence. And then instead of adding DNTPs only, we add additionally a modified form of that, which are called DDNTPs. These are dideoxynucleotide triphosphates. All right. So the trick with the modified form of PCR is you have to add the DNTPs and the DDNTPs in a specific ratio so that as the amplification is occurring, randomly, you will get one of these that will incorporate into the growing strand. This modified DNTP, the DDNTP, is chain terminating. Okay. It lacks the 3' hydroxyl, so you can't add a nucleotide into it. So when your new strand is growing, all right, let's say this gets a regular A, a regular T, a regular C, and then all of a sudden a DDNTPG comes in, it terminates, it stops. So, before you load up your sample into your sequencer, you have to do this type of PCR that uses these modified DDNTPs. Okay. That's dideoxy sequencing. So, up to date, dideoxy sequencing is still like the standard. It's got very good accuracy. It just doesn't have very good read length. Forensic scientists have to do this method because it's reliable. But there are new sequencing techniques that have come out called next-gen sequencing that isn't as accurate. Occasionally you'll get mistakes, but it's a lot faster. You can sequence now like entire organisms, the whole genome, in a matter of days with these new techniques. All right, any questions about that? Good. All right. Okay. So moving on with this section that we were finishing up. So the next part looks at generating recombinant proteins from clone genes. Yeah, I'm sorry. It's random, but so like I was saying, you set up your PCR reaction with a specific ratio of regular DNTPs to those ddNTPs. So it's usually you have less ddNTPs in there. And randomly, because there's so many rounds of replication going on through your PCR, eventually you're going to get a representative population of all of these different strains. But it's a random event. Yeah, it's random. I mean, but you have ddNTPs for the A's, T's, C's, and G's there. So these are all your dNTPs, and then randomly you get incorporation of one of these ddNTPs. And as long as it's ratiometric, you'll get pretty much an equal number of copies of each of those fragments. Okay, so let's say you use PCR to amplify a region of DNA and you want to study it. So a good next step might be to clone it into a vector. And even further than that, let's say you want to make up a lot of protein from that DNA that you put into that vector. So you can do that by using expression vectors. Now, this is specifically talking about prokaryotic expression vectors, because prokaryotic expression vectors use the Shine-Dalgarno sequence. The Shine-Dalgarno sequence is for binding of the mRNA to the ribosome. But it doesn't mean you're stuck with only expressing bacterial proteins, because you can take a eukaryotic gene, a human gene, put it into a plasmid, and then put that plasmid in bacteria and use the bacteria to build up your protein or to make your protein. Okay, so once it makes your protein, you can isolate it from the culture. This is how insulin is made now. It's recombinant insulin. So we're actually using the human gene for insulin, putting it into an expression vector, but using E. coli to make that insulin. All right, so here's our typical expression vector for prokaryotic systems, right? It's got a Shine-Dalgarno sequence, which is necessary for ribosomal binding of the mRNA. It's got an origin of replication. It's got a resistance cassette so that when we culture this, it's in the presence of ampicillin, and only cells that have taken up the plasmid that have been transformed survive. It also has a promoter, which initiates the production of that mRNA, right? Transcription. All right, then we take our cDNA, which is possibly eukaryotic, we clone it in. Remember, we use DNA ligase to do that and restriction enzymes. And then we transform it in bacteria and it produces recombinant protein. All right. I think that's all of 3.3. Oops. Okay, yeah, that's all of 3.3. All right, so 3.4 continues to go into techniques, molecular biology techniques. Again, hopefully a lot of this is review. So techniques like southern blotting, northern blotting, and fluorescent in situ hybridization all use DNA probes as part of the technique, specifically being able to hybridize those probes to your target DNA. Those probes can be radio labeled, either with a radioactive isotope or a fluorescent label, so that you can detect if the target DNA sequence is actually present. Okay. Southern blotting is like, so there's northern blotting, southern blotting, and western blotting. There's no eastern blotting. Western blotting is what we're going to do in our lab, and it involves antibodies, so it doesn't involve DNA hybridization, but southern and northern do. Southern blotting is used for looking at genomic DNA. Northern blotting is used for looking at RNA expression, so the presence of mRNA. The first step in southern blotting is to take a gigantic chromosome, bunch of chromosomes, and cut them up with a restriction enzyme. And so you now have broken down into smaller pieces, and you can separate those out by electrophoresis. And then the next step is to probe it with a radio labeled probe. But you can't bind a probe to DNA that's in a gel. It doesn't work like that. So first you have to transfer the DNA to like a workable substrate. So you assemble this sandwich and you apply a current, the DNA will move out of the gel and it will stick to a piece of paper. We'll just use a piece of filter paper. And it will stick, and now you can actually expose that paper to your DNA probe and it will bind to it. And if it fluoresces, it'll glow, or if it's radioactive, it'll also glow, and you can detect that with X-ray film. Okay. This technique, though, obviously relies heavily on these probes, right? So, after you denature the DNA, is separated out, break those hydrogen bonds, the probe will specifically hybridize to wherever it's complementary to. And now that whole piece of DNA glows. So it's a way of looking for specific sequences. It's similar to PCR, right? It's used for detection, right? But PCR is a little better, actually, because it gives you the code, or it gives you the piece of DNA now to do other things with. This is for simply detection. That's southern blotting. And northern blotting is very similar. It involves transferring it to a membrane as well. But instead of using genomic DNA, you're actually purifying messenger RNA out from cells. And you have to use like a denaturing gel to do that. mRNA is a little more difficult to work with and separate. But you separate out your messenger RNAs, transfer it to a piece of paper, and then you probe it with radio-labeled oligonucleotides as well. The last one is called FISH, or fluorescent in situ hybridization. And this is useful for doing or detecting regions of chromosomes by using these radio-labeled probes. It's nice because you can actually use living cells. So you can do it in a living system, or you can spread the chromosomes out like has been done here. So FISH is done both ways. But you're just exposing the chromosomes to a radio-labeled probe, and the probe will bind to wherever it is, and it will glow. And that's how they're able to make all of these different chromosomes, different colors, able to identify them by their fluorescence. Okay, so that's detection methods using radio-labeled probes. You can also detect molecules using antibodies, right? So antibodies are proteins that contain a couple different regions. Talk a lot about antibodies in this class. They have what's called the FAB region, which is specific against an epitope. Okay, what are epitopes? Epitopes are regions of antigens that the antibody binds to, right? So, these molecules come from the immune systems of animals, right? B white blood cells, or B lymphocytes, produce antibodies in response to the organism getting exposed to a particular antigen. Right? An antigen is any foreign substance to the organism. It can be a toxin. It can be a benign molecule like dust or something like that. You end up having a reaction to that. It can be another protein, it can be a virus, bacteria, anything that's foreign is considered an antigen. But you also have self-antigens as well. Okay, so these molecules that the FAB region of the antibody binds to and whatnot. The FC region is the constant region. It's found on the other area of the antibody. Constant region is more representative of the organism that's producing it. So if you like collected all the antibodies out of your serum, they would all have the same FC region, but the FAB region would vary depending on all the antigens that you've been exposed to in your life. All right, antibodies are divided up into two classes, monoclonal and polyclonal. Okay, so the polyclonals are typically what are produced, mostly in your cells. And what happens with polyclonal is more variability, right? So you'll have, like if the cell is producing polyclonal antibodies, it'll produce a bunch of different antibodies that have slightly different FAB regions to bind to different epitopes, but usually of the same antigen. Okay. And with monoclonal antibodies, so these are made, can make these in the lab using a modified type of B lymphoma. So it's actually a cancer cell, it's a myeloma cell that produces the same exact antibody. All right, so the FAB region is the same, it binds to the same epitope. Monoclonal antibodies are nice because when you're doing immune detection, one big problem that you deal with is non-specific binding of the antibodies. An antibody binds to somewhere you don't want it to stick to, and you're detecting the wrong target. So monoclonal antibodies kind of take that problem out of question because the antibody binds to the same epitope, all the copies of it. But with polyclonal antibodies, there's some benefits. You get kind of amplification of the target signal. You get a little louder signal, but again, you have issues with background. Okay. So these are derived from heavy and light chains. And basically, the heavy chain is the larger portion of the antibody. The light chain is the smaller portion. Both are involved, though, in antigen binding. We can easily generate antibodies by simply injecting animals with the antigen. Right? So they will develop an immune response against that antigen. They will produce antibodies, and then we can purify the antibodies from that organism, from the serum. When you're working with antibodies in the lab, typically the animals that are used for production of these antibodies are mice, rabbits, and goats. These are just the most common. And there are thousands of antibodies commercially available that bind to particular proteins that you may be trying to detect in your experiment. Additionally, with immune molecules, you can identify modified forms of proteins. For example, we've talked about phosphorylation, right? It's a post-translational modification. This is our protein, and it's phosphorylated at a specific residue. You can have an antibody that looks specifically for that phosphorylated form of the protein. So you can probe for samples to see if the protein is actually active or inactive. All right, so the most common antibody-based techniques are Western blotting, which is used to detect a particular protein. Actually, all these are usually used for detecting proteins, but Western blotting is a little modified because after taking all of the proteins out of a cell, you then separate them all based on their molecular weight. All right, immunocytochemistry is used for visualizing a protein like in cultured cells. So you take cells, you put them on a slide, you expose them to the antibody binds to wherever the protein is, and then in that FC region, you usually have some modification that either glows or is radio labeled or that undergoes a chemical reaction. Maybe it precipitates a chemical to give you a blue signal or a red signal so that you can see where the protein is. Wherever the antibody is bound, the protein is going to be bound. You can detect this with a modified form of microscopy called fluorescence microscopy. So you shine, ultraviolet light on it, and because you have that conjugated region right there, it'll glow. Immunohistochemistry is similar to immunocytochemistry, except it's done in whole tissues. So you'll take a tissue slice, put it on a slide, and then expose it to an antibody. So those are the detection techniques. The last one, which is immunoprecipitation, actually allows you to pull the protein that your antibody binds to out of like a slurry of proteins. So let's say we take a mass of cells and we break them open. Now we have what's called a cell lysate. Right? So in our tube, we've got millions of broken up cells, and we have thousands, hundreds of thousands of different proteins in there. And let's say we want to purify one particular protein out of that mix. We can use immunoprecipitation to do that. You add an antibody against the specific protein, and it's going to stick to that specific protein. And now you can centrifuge it. The heavier antibodies will come out first. We'll sediment first. And it's a way to purify your population of a particular type of protein. Okay. So here's Western blotting on the left, also called immunoblotting. So you basically take a lysate, you denature the protein. There's an important chemical in this process called SDS, which is sodium diacetyl sulfate. It's a detergent that coats all of the proteins in a negative charge. It also denatures them, it linearizes them. That's why it's called SDS page. So sodium diacetyl sulfate polyacrylamide gel electrophoresis. And the first step is you run a polyacrylamide gel and you separate out all the proteins that have been denatured by the SDS according to their size. The SDS coats it in a negative charge, like I said, that allows the proteins to migrate through the gel. Put a positive charge on one side, negative on the other, that'll move. And they separate out according to size. But you can't probe the gel with an antibody. It just doesn't bind. So you have to move those proteins to a piece of paper, just like in northern and southern blotting. So we use a piece of nitrocellulose to do that. Right? It's like a polar membrane that proteins will stick to. And now we can probe the piece of paper with the antibody. So on that piece of paper, you have a bunch of invisible proteins. It's been separated out. You add your antibody to it. It'll find the protein, and now we can put some X-ray film over it, and we can see exactly where that protein is on the blot. And there's a little more to that. You have to use 2 different antibodies. We'll talk about that in lab and stuff, but this was introductory. All right, this is immunocytochemistry over here. So you fix the cells. You take your cells that you're going to look at under the microscope, and you fix them. You treat them with paraffinialdehyde or ethanol. And that kind of cross-links things in the cells. It freezes the cells in time. You expose it to your antibodies. It'll go out and find the proteins, and then you blast some light on it to detect those antibodies, and you can see where the proteins are localized in the cell. All right, finally, immunoprecipitation, like I went over before, you can actually sediment out the protein that you're looking for because it's heavier, it's bound to the antibody, and also it's insoluble, right, because it's bound to that larger protein. All right. All right, any questions? Again, a lot of this should be review. Any questions, guys? All right, rather than take a break, I'm just going to kind of jump into this project because it's going to be kind of like a break. You don't have to listen to me talk. So that's a break. So I got sheets you guys are going to grab up here. I'll just pass it. That's one of these. And you guys are going to break up into groups. Just the people around you will work. 3 or 4 people can do it.
Oh. Got you on.
Okay, so when this works, there are 4 or 5. There are like 4 or 5 data sets in here. Right, I've given you all of them so that you can review all of them, but only one of them your group is going to do. So if you guys could, does anyone not in a group here? You guys got your group set up? All right, so I just need you guys to figure out who's going to be the group leader. Right? Group leader is just the person that's going to write the answers down. So preferably someone maybe with good handwriting is good for this. So when you have that figured out, just go ahead and that group leader, if you can raise your hand, and I'll give you a number. Number you're going to do. Number 1, number 2. All right, where's group 3? You guys a group? Group 3. You guys are group 4? I think there's 5 here. Okay, so listen up. This is what you're going to do. Each of these are sample data from a research paper. Okay. Try to pick these as carefully as possible. You should have all the background information that you need to review these questions. Except for group 5, you guys got screwed. We didn't go over chromatin yet, so that's okay. So you might have to do a little reading up in the next lecture, next chapter, stuff, but not too much. Whoever that group leader is, you're going to fill out that first page. So put your members on there, put your group number. Right, you're going to turn this in next week. Actually, maybe I should make it 2 weeks. You guys focus on everything. Okay, you're going to turn this in 2 weeks for a little grade. It's nothing crazy, but on the answer sheet there, you're going to answer the questions for your data set. Okay. It's really important that you do this as much as possible together. Okay, so the group leader's not doing the work. It doesn't work like that. And also, I don't want ChatGPT to do the work either. So when I read these, I guarantee I'm gonna know if you use ChatGPT because I've run them all through ChatGPT and I know the answer that it's going to give you. So the purpose of this is to get you to start working through understanding scientific data. All right. So when you read it and look it over, I guarantee you at first it's going to make little sense to you. But with a little conversation with your group members, looking at it more closely, thinking about the things we have learned, I know it will start to come to light. Okay. So it just takes a little time to work through these. It's not something you're going to spit out the answer in 5 minutes. Occasionally, you're going to need to look up something. All right. If you don't know what C3C protein is, and it doesn't define that, you might have to look that up. Okay. If you don't know what Kamasi Brilliant Blue is, you may have to look that up. And that's okay. I don't want you looking that up. Google's your friend for that. The textbook is your friend for that. But I do want you to challenge yourself to work through the problem. Right. Also, any of these are games for exam. I mean, I'm going to be gentle with that, but this stuff can get pretty difficult. But I want you to be familiar with these data sets that we're going to work through. Okay. All right, so I'll give you guys about 20 minutes or so to get started. You don't have to get finished today, but I do want you to start on it. Work with your group, read through the problem that you're assigned to, look at the questions, start interpreting the data, and then start assembling an answer. I'll give you guys till like 10:45. Okay. 10:50 to get as far as you can with it, okay? Yes, ma'am. Did you, okay. Not right now. I would rather you do this, but, I could probably have you take it after, so.
I have everybody number except your. So I do.
Yeah.
As long as I can figure out. Yeah. I figured out part two system. I single into your mouth. Yeah. Question number three, because you're really good. I can't. That is very true. Yeah. Related to the. Okay. Yeah. It ultimately will see just get to the others, so we'll make sure. Okay, that's everything. Actually, I got mine, we don't have. I would say we didn't say that, because you're, what you guys want to, I can't add you to that one. Some you're not really not, but.
So remember, each group is only submitting one copy of this in 2 weeks, okay? You guys keep these and go over. Yeah. Yeah. Michael, is your phone number still 727? 868. Separation is like, access to the sea. How is it? Hey, I'm gonna give each group an extra copy so that you get one or the other guy. First, let's write it up.
I think we'll be IR.
Yeah.
Yeah.
So, just making sure I'm reading the Jolex first.
Yep.
0.01, that's just decrease the student.
Zero point zero one is the amount of DTE that.
Like, you see, where's the type of
Yeah. So such living and you guys just basically kept the CDC, either CDC.
Yeah, so all, what you're looking at here is just, just some different. I'm not gonna give you the answer. So, how does it happen? One, that's what it has. I think that was just. You guys need an extra copy for. I'm gonna post it on campus.
That's all.
What was that?
We created the exact age.
Oh, sorry.
And it gives them.
Department Chair Kelly. She just wanted me to give you a message to remind you guys that she's always welcome to meet with you. You're always welcome to meet with her to go over your schedules. Like your, as a, in terms of advising and stuff, so. What's that, split it over 2 days? I wish they could do that now. Anyways, a bunch of free stuff back there. She said you guys could have, so it's on that back table. Help yourself to it. This is pens and whatnot. Can we?
Yeah. So, what the exactly?
Well, let's talk about this. I'm ready to That's your major. I don't see them in one like less fun. I'm sure that's the kind of reason. So, that's. I think that the one of the two is most related than the.
One, two, whatever.
You think more outside of the box, it makes sense. Right about the front.
So there's no advance, right? 1 to 5, 7 to 11. You'll see how the charge. It's going to the the top. How that plays into it. It has like a.
Yeah.
It's all directions to go. So. Has to do with this time.
You reason.
Really? Yeah. So you had to turn their down the paper. So.
So actually, this young lady has to really good question. When you're answering your questions, don't just answer the question. I think you should add your reasoning. Why did you choose that answer?
It's.
Yes, ma'am.
What would you want for like a reason?
Power GNA office visualize anything else? So did it say specifically?
It's more of like,
Yeah, so there's different ways to visualize the DNA after agarose gel electrophoresis.
we just wrote about their visualizing for fluorescent dye and stuff.
Yeah.
Like when we have to put like a reasoning for that, because that's covered.
Lithium bromide, right? That's the most common one used. Oh, these quiz grades, guys. Holy shit.
I think 6 is.
So like one or two of them are thicker. Same for but that should, I think that would, that's something that's what you would expect them to, you want to be similar in fragment size as far as their travel, to get close to the 140 line.
But they're broken. With the fragment. So you can, one thing you can see under a reducing agent condition, but the, this is in the reducing. Okay, so. This is after you. Or. Excuse me, and so the things regularly, as so you can differentiate. So I will use it again, what is visually this, break it up, get rid of the balls. Yeah, you're yeah, you're just getting these individual aspects, right? So the more you can have, the more cutting out. Right. Okay, we did some from my giving place. The think it was some colors. So there we go. Yes. So then you have them color next to it, right? Yeah. One of the there's three. But I don't know where it's all the rest and keep it in. So you can see is. Then it stands proteins that have been fitted to. By DT.
I think it's.
These are all.
Migrate visual.
I'm assuming it's migrating. No, I think it's.
Smaller, but larger, migrate.
Bottom of it.
It's gonna be the same. If you, I think. Yeah.
But I feel like once you get so many, the more professional, less. And
Which is what they are.
So,
It's actually. So it's then.
There's so much.
Either. Prime or. No first.
Yes, ma'am.
Okay,
So that's probably one.
are you gonna be hearing for correctness? Trying,
Both.
because I feel like I'm just talking gibberish.
Which one are you doing?
Number two,
Yeah, so this one has to do with, so go in your textbook and look up the figure that shows how the glycosidic bond is made. So that'll help you a little bit.
okay,
Key enzymes, spartans, carbolase, Yeah. Yeah, look up that and then also consider like enzyme activity, right?
Okay.
How when you're adding more substrate, what happens in the reaction? Look at specifically what you're looking at here. You got to understand these figures in order to understand the question.
so is ATC an allosteric enzyme?
ATC, activity of ATC is this. It's an enzyme.
Okay, but is it an allosteric enzyme?
Why does that matter?
I don't know, because doesn't that decrease the activity?
Just think of it as an enzyme here in this problem. Think of it as.
Everything is the same. And then 75.
Yeah, keep thinking about it. So
75.
all the like principles in here are relatively simple. Like they're just set like enzyme kinetics and substrates and products, but it's a lot of different principles all coming together in one figure. That's what's making it tough here. So,
You
dissect
said
it.
look up,
Yeah, if you look up like the UDP or how the glycosidic bond is produced, that might give you some insight. Yeah, so be sure you're reading this carefully. You got 3 sets of reaction mixtures that were set up. Each contain ATC, these things. Those are the 3 mixtures there. So just keep looking at it. I want it to come to you after you've been looking at it for a while, and it will. Why are so many people absent today? All right, that sounds good to me. That usually has little effect. That's all about. A lot of zeros on this quiz, though.
That's so loud.
Yeah.
All right, guys, it's gonna give you like 5 more minutes. I know you're not gonna get done. That's okay. No 2 weeks.
That works. So far, all we know that on the place I do that film is working. We're working for, based off of what's happening. It's trying to. But it's the same what is NTV. I've been literally, what is NTV? It's not too far.
Yeah, it is.
Oh my gosh. Still the Lord. I think we get.
Oh, it's just. And we did school stuff.
Again, for you.
How is the one of the trees?
Something.
Oh, what's this?
Yeah, it's a fragment of the,
There's a book note too, that's very.
It says how many times.
Yes.
I'm sorry, give me two. Are you sure about I'm gonna say, I don't like. If I can look at it, it's great.
Yeah. There's 102 races.
Yeah.
75. Yeah, okay. What is this?
Yeah.
Okay. Yeah, look for I'm stupid. Where are you seeing this?
oh.
In. But you can actually compare it to right now. Because I see sometimes that. Yes. So it's. All right, and then it won't. So we got one or two. How did we? Work it out. But I'm excited or something. That's what's, that's me, that's not, no, it's not Christmas. You probably would get. Oh, we have self-viable. Yes.
So you got.
Specifically, brand that I see from that one.
I was like. You know, that's over. Yeah, okay. I mean, no,
Easy to the right.
so like. See. But it says determine their next molecular mass survival. 75, 43. 20 that. Set that is to the province of the.
They should know that.
Okay, so.
Oh, from today?
They have this.
So how many answers to your question?
You're on the right track.
Well, there
What was
is,
the question?
yeah, and one of us.
You guys are on the right track.
No, you're right. Happy top.
Either right or take it.
Okay, yeah.
All right, guys, couple more minutes here.
But we don't have. No, we have no person willing to submit it.
No.
Yeah, no, I was trying to do that.
We're just doing the first one.
What's easy?
There we go. No, that's great. You gotta go silent. So you're doing that. Yeah. Either way, we're doing everything in the packet.
You should look over everything, yeah, you should answer all of them, but you only need to turn in the answers for your assigned thing, and you don't need to turn it in today.
Yeah. The rest of your life, now you can look at them today.
Yes. One could assume. That this is. So how do you know it's not?
Your name, you know, it says on it already, but it's just for me, it's all. Nothing.
Yeah, meaning we have to.
Yeah, I'm gonna start randomly, it's actually good, all right, hey, let's just get there. Yes, they were in nature. What is the other names? So one of seven, PPR is, what are the PPR result statements? Yeah, result statements, what I struggle with. But it's we last year.
We're running out of days.
Yeah.
Just match it up all. Okay, so yeah. That's what I'm saying. This is like. There's a, so I don't three, but there's that's right. That's the bit of the last night. Yeah. Yes, it is. So, it'll be fine. And the other time, so then I'm just. If you spend one, so I said, like one, violent time, so that's one thing, right, one that doesn't have enough. I don't put it through. It's been a but once I remember you. I'm just saying, you know. Where's the dog? Oh, I don't know something about doing it again. Yeah, they want to see that. It's so. I think it's me. Hey. See, like, flat back. You think you're gonna do that? That's Monday? Everybody goes there. They'll be there to fast. Just what's around here? What? That's what Sources to go and see it. Program. Oh, sorry, God, but. Okay. Just taking some photos tonight. Where's the? Oh. I ain't going to Nova Zoo. Yeah. 0.25.
Yeah.
I did for me, I want to see. We're going to crush it. Oh, school. It's not. Else you stay. The one that's over, you see like another big. Oh. And then there's Kansas State and Hungary. Check out. There's a pretty beautiful. They don't have too much. No, they didn't. Ohio didn't have it. Exactly. Yeah. Might be into that, but yeah. Wanna hop on one of the horses? They said, I'll be fine. I know how to ride. That's cool. Yeah. Get my way out of town. I got it. Yeah. And I run by with my aunt's girls.
If you want.
I would like to.
Can you run?
No,
Okay, well, I didn't know if you had a copy. Did you get to see it? Or-
probably. Yeah, why don't you come up here?
Okay, so I will do that.
Okay,
So
so,
this is the reducing agent on protein structure. What's question number 4?
so number 4, that's all mumble jumble, but pretty much I just wanted to confirm that relative to the other, species there, is it trying to indicate how like 102 is a segment 102 and 43 of 145 that have split and then traveled based on their molecular weight? Is this all asking in reference? Because 145 is the molecular weight of the C3C protein it's been talking about.
Okay, what is the, what'd you say it was, 103 or something?
105 is the molecular weight of
Yeah,
what's comparing.
Pretty close to 102.
Right. But is that what it's asking? I'm making sure I'm not, I just think more outside of the box. Like it's all relative to this initial one that we're looking at.
Yeah.
And then how it traveled once it was denatured. That gives us the size. They're all fragments of it.
They're not fragments. 'Cause
Yeah,
It's
how
it's
would I
still one
rephrase it?
protein. It's just,
Okay.
it's boiled up differently. Like, so when you add a lot of denaturing agent,
Mhm.
right? It really unravels the pieces very well, so that it's completely linear. But when it's folded up, it'll migrate faster. That's the whole thing here. So, electrophoresis will separate fragments based on size if all the sizes are in the same conformation. But if the DNA or the whatever you're looking at is this protein, this is protein.
Yeah.
If it's not completely denatured, right, it'll coil up and it'll actually migrate faster than the whole linear. That's the whole concept.
Oh.
Okay, does that make sense? Does
Down.
that make sense?
Oh.
Okay. All right, 5 more minutes. I know I keep saying that, but I really want to keep talking.
So then that should tell me that this is in its linear form. That's why it hasn't.
Well, so
traveled.
the linear form, I would say is probably going to be around 10. Yeah, I mean 105. That should probably, it's probably linear form. This could be possibly dimerized together. It could be stuck together.
Okay, so 102 is likely in the linear form, so relative to the other ones, these have probably been coiled up. And that's why they're traveling further.
Yeah, and just be careful of that terminology. So coiling more has to do with nucleic acids. They coil. That is, it's just not fully denatured. Just with the terminology. But you are on the right track.
If it is fully denatured, it should be,
Should be representative of its molecular weight. It should migrate according to its molecular weight.
Okay, so you say so. Basically, wait, how do you have? Most likely, it's not here. We're only comparing everything to the 105, right? They are. It's up here, the guys are C3C. So this is probably in a finger form, and then, saying to be careful with this terminology, but the rest of it is likely closed up. That's why it's migrate faster. That's why we see that here. So that would be another comparison that would be, as far as it relates to the other species. So one is really relative to the others. And that these are probably like tied together or something like, because it's what we're comparing to. So like, that's just always gonna be up there for 145, like it says a thing. So essentially just more like. Which has everything to do though with the deep. So whether or not it completely brings it down or not, or being perfect. So we're right. Oh, God. I'm staring at you, so I'm being. Go for it. Okay, so.
Or you don't need to be. What's the energy or best way some shoots there's a what's like side of sign.
Yes. So is this like a circular thing? Is this happening? As long as you have. All this one.
It's this one right here. So it hasn't, this primary, okay. And this reason, yes.
Well, it is full. So how about? So we did not make CPR, that doesn't work. I think we're going a little too.
Yeah.
Okay, so
A negative.
that's the product, yes.
We have the product, if we make too much of it, we explode just, okay, so that's why we want to inhibit it. Okay, I'm saying, if we burn off that, then we keep that, we can see, so then we're not gonna end up getting too much like flow into the surface.
Yes, it's negative feedback. It's not just
It's doing
stop that.
Yeah, it's negative feedback signals, the animal instead of that.
Okay, so this. By the collective racing. That was. Yes. By. He said. But then I wonder if it's. This is a more, either maybe there's something that's great. Or it's too funny. Sourcing the.
Yeah. Don't get for us.
If it's coiled up, sort of, and then more or less.
Well, it's less denatured. Like fully denatured means it's been fully linearized,
Right.
right? You have to also like what these DTT specifically, what sort of bonds are it breaking, right? So, think about that. That's an important component.
Okay, that's only step one. All right, part two. Question one. Is it safe or it's actually brand new? Oh, we're gonna do that. So I like to bring that machine up. That's gonna be this kind of exponential. Yeah.
I ended up writing, but it's like whatever. So you weren't listening, right?
I was not good. I did my runs early before me.
So it's
Okay, together. I think that's it. I'm aggressive to then. I don't even know. This is more complicated. And overproduction. Well, I'm glad you're looking. This is farm. I'm looking at different green, but that was actually. It's like.
she was getting on. I was like wearing shorts.
So. Right. So I just said. I mean.
She's ever taught this course.
It's
very good.
She did say it was first.
Yeah, she was gonna the direction.
That's what he did. He was like,
I just kept forgetting stuff. It's like literally birds are like popping up.
One.
Literally.
Oh,
Flash. That's what she did. Walk out to the detention pond. Sediment, water.
Yeah, but that's it, yeah. So when this one goes, this one then can go. And the insurance. Why is there so much? Which container, Peter end up signing out of these containers, so why can't they sell me?
all right, guys. Go ahead and put those away. Got more stuff to cover.
Okay. But I thought it was supposed to go to positive. Is it upside down? Because it's supposed to go that state. So driving the wheel. That's something a little. Yeah, but it'll be all down. Everybody got back in.
All right, so I think what we're gonna do, we're just gonna cover up section 4.1 today. I'm gonna do 4.2. Be a little less, so, but like I said, whatever we don't cover today won't be on the exam, so whatever we cover up to. Technique, study gene, this is where we're at, right? All right, so that seemed to go pretty well. You guys did good. Start to figure that out. So just remember, turn in your answers 2 weeks from now. And we'll be all good with that. Okay, so in the last chapter, we were talking about prokaryotic expression vectors, right? So, if you want to mass produce, bulk produce a protein, a prokaryotic expression vector is very useful to do that. Also, it's very cost effective to do it that way. Okay. Bacteria are easy to grow. They grow in pretty much just chicken broth, and at a simplest level, you can grow a lot of bacteria in that. But oftentimes, you need to express proteins in mammalian cells, especially if you're studying the effect that protein has on the cell, such as the signaling or something like that. So mammalian expression can readily be done in cells in culture, but it can be done with modified versions where you're actually expressing the protein in whole animals as well. We'll talk about that. So if you're doing cell culture, though, you've got a whole different, from mammalian cell culture, it's a whole different system to be able to grow those cells. They grow very differently. They require much more specified and fine-tuned growth medium. And often that growth medium, can't be produced completely artificially. It's just too complex. You actually have to supplement it with serum from a living organism. So the cost to do mammalian protein expression is pretty high. You can't grow these large vats of cells, although there are systems to do that, but it's very expensive. Okay. But in basic bench research, it's invaluable to be able to put protein or use mammalian cells to express particular proteins. Okay, so that's what this has to do with. Now, the process of introducing foreign DNA into bacteria, it's generally termed transformation, right? You've transformed the bacteria. When we're putting DNA into eukaryotic cells, we call that transfection, right? So if you see those terms, usually transfection has to do with eukaryotes. Transformation is most commonly assimilated with prokaryotic introduction of DNA. So you have a culture dish and you're growing your mammalian cells, and most cell lines that are grown mammalian are adherent, which means they stick to the bottom of the dish. Some of the blood cell lines are suspension, they kind of grow in solution. And you're growing it in your specialized incubator, you got your special media in there, and then you dump some plasmid DNA onto it. All right, so cells will naturally take up exogenous DNA. So they will take up those plasmids just through endocytosis, right? The plasma membrane kind of invaginating it in and pulling in the plasmid DNA. So if you have in your mammalian expression vector all of the components that are needed to drive expression in a eukaryotic cell, you will end up driving expression of your protein. Okay, so the expression vectors don't use that Shine-Dalgarno sequence. So we'll use a different sequence, like a Kozak sequence. And you can't make the cell resistant to an antibiotic because typical antibiotic isn't going to kill a mammalian cell anyway. So it's different I don't know why they have ampicillin there. That's not right. So it's different drugs that you would use to select for the mammalian cells that have your plasmid DNA. Neomycin is one. Use neomycin. All right. So you put your plasmid DNA in, and the cell incorporates it. It starts producing the protein or the gene that is in your plasmid. And maybe the cell doubles once and it produces a little more protein, but that's about as far as it goes, right? We call this transient expression, right? So the cells are producing a little bit of protein, small amount, maybe only after a couple generations of division. Oftentimes, what you want is to produce a cell line that stably expresses a protein of interest. So the reason that the cells are transiently expressing it is because the plasmid has been taken up. Here's your nucleus, and it's in the cytoplasm. Right. So, transcription typically occurs where? In the nucleus, right? So these things can like go in and out, but they're predominantly in the cytoplasm. And so you get relatively low expression, and it doesn't last over generations because as that cell divides, you start to lose the plasmids over successive generations. However, as a very rare event, that gene will pop out of the plasmid and actually incorporate into the host genome. And when this occurs, you've created a stable cell line. And as long as that, for as many times as that cell divides out, that gene will still be present in the genome of the cell. Okay. So, this requires a lot of selection to do because it's a rare event. Like maybe only one cell in your population of millions might actually do it. It's much less than 1%. Yeah.
Is there no way to like mechanically force the cells to incorporate?
There are ways to like instigate it to help it, but mechanically, I don't know of anything mechanically. There are a lot of we're going to talk a little bit about it today with transposons, like mechanisms for the cells to actually shuttle pieces of DNA in it. And then there are things you can do to the actual plasmid DNA, like linearize it, things like that, which maximize the incorporation into the genome. But yeah, physically, moving it in there, no, there's no way to do that. It's a pretty random event. Okay, so these expression plasmids, similar to prokaryotic expression plasmids, but with some subtle differences, right? Obviously, you have to have an origin of replication that's specific for mammalian systems. You have to have a promoter that will work in mammalian systems. Viral promoters are great if you want to drive a really high expression level, things like that. Okay, but you can do the same thing that we did in bacteria in mammalian cells. And you can select for those cells in the same manner that we, use selection agents in prokaryotes as well. Okay, as long as the plasmid confers resistance to a particular compound, you can do that. So, generating cell lines that can be used as models for a particular disease is often a very popular result from generating stable clonal cell lines. Okay, so here we've got, this is just what I was talking about. You've got your cells that are kind of in a monolayer on your petri dish. They're growing there. You dump your plasmid on it. And there's a few different ways that you can kind of instigate the uptake of that DNA. But only a few cells take it up. So you add some drug to it. Only the cells that are resistant to it will survive. And they start to grow out. And a very rare event of those cells that took up plasmid would actually be incorporation of that gene into the host chromosome. But it does happen. So how do you actually get the DNA in the cell? Well, cells will take up exogenous DNA, but they don't do it very efficiently. And usually to do transfection, you need to get a lot of DNA. It's actually like micrograms of DNA that we're dumping onto these cells, pretty large amount. And so one way you can get it in is injecting it in. We have micro injection systems that, take these glass, basically you take a Pasteur pipette and you heat it, you stretch it out, and it makes a really, really small needle. You break it, and then you can use like a robotic guider, either robotic or manual, that can, hold the needle very stationary. You have the dish very stationary, using like an anti-vibration table, everything, keeping all that. And under the microscope, you can actually inject the cell and then, using a very guided air pump, push the DNA into the cell. You can also electroporate cells. So here you take cells, you cool them down, and you put them in what's called a cuvette. And the cuvette has these leads that you can conduct a current through the solution that contains your cell. And when you it's like some Frankenstein stuff, when you electrocute the cells, it kind of creates pores in the membranes that allows the DNA to fall in. You can also use calcium phosphate. So calcium phosphate will precipitate the DNA. It pulls it out of solution, so it settles on the membranes of the cells, and that instigates endocytosis a little more. So it'll bring the DNA in. You can also mix your DNA with what are called cationic lipids. So these are lipids, have these long fatty acid chains, and they have like an anionic region. And so the DNA will kind of stick here. But because it has these lipid regions, those lipids will associate with the plasma membrane, bringing the DNA closer to the plasma membrane and instigating endocytosis. All of these are very commonly done. Just depends what you're kind of doing and how much viability, there's different toxicities with these methods. And some cell lines, mammalian cell lines that you may work with are very delicate. Like they're really sensitive to some of these methods. So you just have to try different ones. The last one is more biological and involves viral delivery to the cell. This is a very high efficiency, low toxicity method to do it. But, you're working with virus. So, if you're introducing an oncogene, which is a gene that would make the cell cancerous, into a mammalian cell, and you're working with that virus in the lab, potentially that's pretty dangerous. But these viruses are bioengineered to be replication deficient. So when you get infected with it, it's not going to replicate. It's not going to be infectious. Yeah. Uh-huh. Endocytosis. Most of these, like both chemical methods induce endocytosis, which is the membrane kind of caving in, and there's your DNA. And eventually that becomes what's called like a liposome. So viral delivery, though, the virus actually attaches to the cell membrane and squirts the DNA in. And you can do this very artificially in the lab. So take a tube and you mix in 3 plasmids that code for everything you need to build the virus. They're abbreviated GAG, pol, and ENV. So the envelope, the polymerase, and the GAG sequence, which will all assemble the virus. You mix those in with your plasmid that has your gene, and you mix it up, and then you infect cells, and the cells will produce the virus just from these 3 DNA fragments, 3 plasmids. So now you've got virus in the media. You take that media and you expose it to the cells that you want to transduct or transfect. And that's one thing about viral mediated delivery, they call it transduction. These other methods are more transfection methods, just terminology. Adenovirus, retroviruses are really commonly used. All right, so they use these retroviral vectors to assemble that virus. This would be called a packaging cell line because it's taking those components to build the virus, packaging it, building the virus for you. And then it gets in the media and you expose that media to your target cells. That's what's happening here. You've got the different plasmids, you're assembling it here in a packaging cell line, and then you're exposing it to that media to an additional cell line to actually get the DNA. A big benefit of this, and like you were asking before, what are ways that you can maximize the incorporation into the host genome? And this is one of them. That's one thing about retroviruses. So retroviruses, when they infect, oftentimes their DNA, right, will get incorporated in the genome, and that's why you can have lifelong infections from these. If you get cold sores, right, that's actually caused by a retroviral infection that exists on the optic nerve of your eye. These cells are always harboring this because the retroviral sequences are in the neurons, like in the nucleus of the neurons. Retroviral infection will maximize your target gene getting into the target cells. It just increases the efficiency so that you produce a higher rate of clonal stable cell lines as opposed to just that transient transfection. Another benefit of viral delivery. Okay, so what we've talked about so far though is, doing gene expression in prokaryotic systems and doing them in cell culture systems, right? But taking that enormously further, you could actually produce organisms that are genetically modified. And to do that, you are doing germline modification, right? So of all the cells in the body, you've got your somatic cells, which are all over your body. And then you have your germline cells, which are the gametes, right? These become the sperm and the egg. So if you genetically modify the gametes and then use those gametes for fertilization, that progeny is going to be genetically modified. Okay. One way to generate transgenic mice is doing microinjection of your DNA into the nucleus of a fertilized egg. So fertilized eggs will contain two pronuclei, right? So they've got one from the sperm and one from the original egg. And using microinjection, you can inject it right into the nucleus. And you can take that fertilized egg and you can implant it in a carrier, right? So the foster mother, call it here. And that foster mother will give rise to offspring where some of the offspring are transgenic, right? They're carrying that mutant form that you've put into it. This is all, well, not all, but most mammalian transgenic organisms have to be produced this way using microinjection. If you're doing some simpler model organisms like C. elegans, you don't have to do this so complicated, although you can. There are other ways of getting the DNA into the cell. You can actually soak it, things like that. Okay. So that's producing clones. It's like Star Wars, right? What are ways that you can actually change the DNA sequence in the genome? All right, what are ways that you can mutate a gene in vitro? So you can use a modified form of PCR to do this as well. And this is, basically it's called mutagenesis or PCR-directed mutagenesis. If you remember when we were talking about PCR, PCR involves the denaturing of DNA and then the addition of two sets of primers, right? We have a forward primer and a reverse primer. And the way we've learned it so far is those primers are exact complementary sequences of the region that's going to bind to. And then this area gets amplified. Well, what if you were to introduce a change in the primer sequence? If you were to look at this and blow it up, let's say, all of these are complementary, but right here we've added an A, if not in that sequence. So, this isn't going to bind perfectly to the template strand. It's going to bulge out there where the A is. But when DNA polymerase comes along and starts replicating this DNA, it's going to keep that mutated nucleotide in it. It's going to keep that A in it. So after your first round of PCR, you produce a hybrid strand, right? You've got your original template and you've got that extra nucleotide in there. And then as you do successive rounds, now you have double strands that are retaining that mutation that you introduced with your primer. Okay, so you can see here, here's your primer, which is the orange thing. And it's got a different nucleotide there, one that is not complementary to T here. Right? So it's actually over successive rounds of amplification. Now you're adding that extra nucleotide. It's called site-directed mutagenesis. And it's site-directed because it's very specific. Site-directed mutagenesis because you're mutating at a very specific location in the sequence. It's all working by using mutated forms of the primer that you're synthetically generating. You're using a machine to generate these primers. You can direct it to produce any sequence you want. Obviously, to do this, though, you need to have knowledge of the sequence that you're mutating. So you would have to have the sequence of the gene that you want to change. But it's very efficient, effective, and relatively easy to do. Okay, as it goes through all the PCR rounds, now you have consistent copies of it that are all the same that retain that mutation. Okay. Again, these are all different techniques that can be done to study the function of genes. All right, let's say we want to see where a protein is located in the cell, okay? But we don't have an antibody against it, or we want to look at something like a little more closely in the cell, exactly where it's located, and we don't want to use antibodies to do that. There's another way to do it. You can create fluorescent fusion proteins to do that. So to do that, you're going to use a specialized vector got your origin replication, it's got your kanamycin resistance, which is for mammalian cells. And it's got a multiple cloning site. That's where we're going to put our gene in. And then after it, it has the sequence for a fluorescent protein. Let's say GFP, which is green fluorescent protein, comes from the jellyfish. And when you when you expose it to UV light, it glows green. So if we put the gene in here for actin, actin is the cytoskeletal protein. When we take that plasmid, let's redraw it. Here is GFP. Now here is actin. We take this plasmid and we put it into human cells. When the cellular machinery starts to do this transcription and translation, here's the promoter. It's going to build one mRNA that contains both of these sequences. That's going to become a single protein that contains both of those proteins in it. That's why we call it a chimeric protein. The chimera, that's like an old mythological creature that had two different heads or something like that. So chimeric fusion proteins can be produced. Now, why is that relevant? Well, wherever actin is in the cell, it's going to glow green. And you can make cell lines that stably express this. So you have like a working tool where you always have, identifiable actin under a fluorescent microscope. Typically, the way that epitopes, right? Different domains of proteins are located on the protein. You may have to maximize which side the GFP is on. So oftentimes these cloning vectors enable you to put GFP on the N-terminal side or on the C-terminal side. And you just have to experiment to see which side will the protein, retain its function, retain its localization, as long as it still works with that GFP tagged onto it. So given the choice, you can put it on different ends of the target protein. All right, so using these fluorescent molecules, yes, sir. You can look at the sequence. You can run it through like protein analysis software. Obviously, if there's like some important important domain here, you don't want to put the GFP on that side. You probably want to put it on this. But oftentimes you have to just produce both the C term and the N term, and then see which one works.
Is it always gonna be the C term and then like the C term first and then the N term, or can it be the N term be first?
Well, so these expression vectors will have, they'll have it like this. Your GFP will be here, and then you'll have multiple cloning site on both sides. And each of them will have unique restriction sites so that you can do it in this, either one of those orientations. Now you have your GFP on either C term or N term. So, localization is not the only thing you can do with these fluorescent molecules. There have been other applications that have been developed that use the fluorescence ability of these specialized molecules. So just something real quick about fluorescence. These are fluorescent proteins that we're talking about, not like fluorescent dyes. And fluorescent proteins, like GFP is the most common one. It's got this beta barrel structure, and it has this fluorophore on the inside. And you're dealing with properties of light. And the first property of light we're dealing with is called excitation light. It emits at a certain wavelength. For GFP, I think it's like 488 nanometers. So this part of the protein pulls in that light, it absorbs that light. And the whole thing about fluorescent proteins is then they emit light at a different wavelength. So the light is now like 512 nanometers. So it shifts in color and intensity. And that's why you can detect it on a fluorescent scope. If it emitted at the same wavelength, you wouldn't be able to see anything because it would just be, mixing in with the background light. All right, so this is how the fusion proteins are generated. I just kind of went over that. You can put it on the C term or the N term. It'll usually put in like a little linker string of amino acids, kind of keep them a little separated so they're not messing each other up. But ideally, these retain the protein properties of both proteins, that you have, a protein now that you can see where it is in the cell. Okay, so that's where I was getting to. So there are two additional applications that you can do with fluorescent molecule. One is called FRAP. The other is called FRET. FRAP stands for fluorescent recovery after photo bleaching. And FRET is fluorescent resonance energy transfer. So in FRAP, it's going to go right to the figure. So FRAP, use FRAP to see basically like how proteins move within the cell, right? So when you shine that emission light on the fluorescent protein, if you keep shining it, eventually it's going to leach out the protein. The protein's going to stop emitting light. Right? There's only so much, we can only do it for so long. So photobleaching is a problem with these fluorescent molecules. You can't keep the light shining on it. You have to do it in short bursts. But the photobleaching can also be used to see how proteins are moving in the cell, because when you use a laser to bleach a certain spot, how quickly that spot fills back up with fluorescent proteins can indicate how the protein is migrating within the cell. And that's what FRAP is. It's used to detect protein movement within the cell. FRET, on the other hand, is able to determine if proteins interact with each other. Okay. So let's say you have generate two different fusion proteins. Okay? This is protein 1 and this is protein 2. And they have not the same GFP, but they have different variants of GFP because there are dozens of these fluorescent proteins that are available. One guy got the Nobel Prize like a decade ago for taking GFP and he modified it using site-directed mutagenesis. He modified it to all these different forms, and now we've got yellow fluorescent protein, red fluorescent protein. He was able to enhance them to make them brighter and last longer so they don't bleach out as much. But we also get fluorescent proteins from corals and things like that. It's not just the jellyfish. So if you have two different proteins and you give the first one the correct wavelength to excite it, it's going to emit a particular wavelength. And if the second protein responds to that particular wavelength, it will then emit its light. Okay, so you're basically using the emission light of the first one as the excitation light of the second one. If that works, you know you've got these proteins interacting with each other. And this is what FRET is. And FRET's a little more difficult. I mean, you need a little higher end of a higher grade microscope. Typically you use something called a confocal microscope to do this. But you can get away with doing both of the FRAP in just a standard fluorescent microscope. Okay, confocal microscopes have like an enhancement system in there to get really, really detailed images of cells. And a lot more sensitive. They use a lot higher end lasers and things like that. Does this make sense? It's gonna be a little tricky to people.
So you rather just chain a bunch of different fluorescent proteins together?
You could. I would get really difficult. But I mean, those proteins would have to be interacting. And I'm sure that's been done. You know, another use of these fluorescent proteins, and we'll talk about it later, is FACS, right? Fluorescent activated cell sorting. And oftentimes, these higher end cell sorters will use 6 of these fluorescent molecules, and you got to sort all that shit out on a screen. It's very difficult to do. But yeah, I mean, if the proteins were interacting and you had the right series of fluorescent molecules to emit, excite, you could theoretically do that. Okay. Cells have ways, this is other ways of introducing mutations into genes and cells. And it falls in like this family of techniques called homologous recombination. All right, we're going to learn about some examples of homologous recombination later when we talk about the development of antibodies and JBD recombination. But there's a lot of different ways to do homologous recombination. During meiosis, simple crossing over is an example of homologous recombination. It's just rearranging pieces of DNA on the chromosome so that you can increase the genetic variability, right? All right, so one of the potential outcomes of doing homologous recombination, though, are knock-in and knockout models, right? If you really want to know how a gene works, having a cell line where the gene has been removed and also having a cell line where the gene is being highly expressed, right, is a really useful tool. If you want to check phenotypes and stuff, now you have a model that doesn't have, is not expressing that protein, but you have a model that's expressing at a really high rate. You can just compare those and see the difference that it makes. So you can also use homologous recombination when working with embryonic stem cells, right? So that's what this figure here. So here you've got, this is like generic homologous recombination over here. Right? So inactivating the gene by it. So you've got introduction of DNA where you've inactivated the gene by introducing a mutation. Okay, so that gene gets in the nucleus, and then through a very rare event, it ends up getting incorporated into the DNA. So here you've used homologous recombination to replace a particular gene with your mutant form that doesn't work. And that's a good way to produce a knockout clone. Right? When you're using embryonic stem cells, right, you can introduce one inactive mutant copy, right? And now it's only got one copy of the homologous pair containing your mutant. But you could also take that organism then and breed it with another organism that has only one copy. Right? So mate them together and it would produce a homozygous organism. Okay, so this is a very it's very arduous to do it this way, but it's highly effective, right? It completely replaces the gene that you're working with, or it completely knocks it out, right? It's not like doing siRNA or something that's very transient in a cell system that only silences something 90%. This knocks out the gene completely. Okay, so that was all targeting the gene in the genome. But other ways to study gene function are to target the transcript, right? So what gets produced after transcription from the genomic DNA, which is the mRNA. All right, so one way to do that is the use of antisense nucleotides. So these are sequences of nucleotides that are complementary to the messenger RNA. RNAi is a very good example of this, specifically double-stranded RNAi or dsRNA. So it uses a short double-stranded sequence, right, that will bind to the mRNA and basically targets it for degradation. It's very easy to do in C. elegans because it's very easy to introduce that double-stranded DNA to it. In fact, you could just feed it to them and it will work. It's a pretty robust effect, and you can actually create mutant worms that look mutagenic, like they look different, by this method. Then it was discovered in C. elegans. So this also discerned this whole molecular mechanism for RNAi or RNA interference. And there are a few steps. So this is just basically using antisense RNA or DNA to silence a gene. You can see, mRNA to protein, but if you introduce a complementary strand to that mRNA, you don't get translation. So it's inhibiting translation. Whoops, I'm missing a figure here. Okay. Oh, okay. So if you're looking in your copy of the slides, it actually goes a little bit into the mechanism, but the next section explains the mechanism in more detail of RNAi. So I'm just going to cover it in the next section. All right, moving on here. Chapter 4. What time is this class over? 11:15? I mean, 12:15? 12:30? Nice stuff. Okay, chapter 4 is on the structure of eukaryotic genes. So when I took this class, 30 years ago, it's changed a lot. The subject has changed a lot. Back then, it was really, you had protein encoding genes, right? You had, the genes that are going to produce the mRNAs and then the proteins. And then you had all this other stuff that made up a majority of the genome that has a lot of function that we don't know, right? But it's that whole central dogma from DNA to RNA to protein that really defines the cell. And what we have found is all that is completely untrue. The structure of the eukaryotic gene is very complex, and that protein coding portion is just a small piece of this enormous puzzle. Remember, all of the somatic cells in your body have the same DNA in it. You have the same nucleus. You have the same genetic instruction booklet. So why is a liver cell and a cardiomyocyte a cardiomyocyte? Why are those cells different? It's to do with the expression of those genes from the nucleus to the ribosome and all that. Obviously, in liver cells, some genes are on, in the cardiomyocyte, some other genes are on, and some genes are off. So the decisions that cells make to decide which genes are on or off, and also at which level to express those genes, is what defines the cell, makes cells different, and creates tissues, and ultimately more complex organisms. So the more complex the organism, essentially, the more important these non-coding regions of DNA are. And also, the more abundant all of that non-coding DNA is in that particular organism. All right. One thing you're gonna not like about reading this chapter is it's a lot of statistics and 90% of these 5.5 KB fragments. You don't need to memorize those, obviously, but there are some important values that I would like you to start getting familiar with, right? Obviously, we know how many chromosomes we have, right? How many? 46, right? Knowing that there's like 20,000 protein coding genes, right? Some of these values can get important. And as we go through, I'm going to try to drive those home, because as you're reading, when you know those values, you will be able to understand what you're reading a little better. All right, but genome size, chromosome number has nothing to do with complexity. Now, is genome size and chromosome number the same thing? Right, genome size should be the number of base pairs. Obviously, if chromosomes are longer or shorter, you can change that. But there's no direct correlation either between genome size or chromosome number and genome complexity. Okay. We know that there is a lot of non-coding DNA. That means DNA that's not going to become a protein. We have, and a majority of the important components to that make up regulatory sequences, right? So the regulatory sequence is what a lot of this course is going to focus on, and how that regulates transcription and whatnot. You'll see that a lot of this non-coding DNA is just repetitive DNA. All these things have implications in both evolution, and you can see ties like between repetitive sequences and different organisms, and that's often used to dictate, classes and species and how things are related. All right, in general, prokaryotic genomes are very compact. Okay? Eukaryotic genomes contain far more regulatory sequences, and that makes sense because eukaryotic genomes contain more different cell types because they contain more different tissues. All right, so back when I took this course, the definition of a gene was something that codes for a protein. That is not the definition of a gene anymore. The definition of a gene is something that yields a functional product, right? Because the genes produce ribosomal RNAs, transfer RNAs, proteins, and other things. They produce microRNAs, picoRNAs. Anything that produces a functional product is a gene. Regions of these genes have different purposes. Some of the regions are actually, used to produce that protein, and some are not. Okay? So they have what are called coding sequences, which we call exons, right? These are starting to become very important terms. And we have non-coding sequences, which don't become proteins, called introns. But both are transcribed, right? So here's your chromosome. This is gene X. RNA polymerase transcribes this whole gene, right, to make mRNA. And within that whole gene, it retains the introns and the exons. Right, so the introns and exons are found in both the DNA and the RNA. And then there is a process that modifies the RNA. In fact, there are 3 important modifications that occur to that mRNA. And I have a whole chapter on them, I think. You get the addition of a tail, you get the addition of a cap, and you also get something that occurs called splicing. RNA splicing is the removal of these introns and the recombining of these exons into a final mature mRNA molecule that can then go to the ribosome and be read to produce a protein. Most human genes, for example, are 90% intronic. Right? So you have these large gene sequences, and most of the stuff is completely taken out. A lot of it has regulatory function. Some of it is just, evolutionary remnants from just hasn't been phased out yet. Take this thing out here. It's annoying. Yeah.
Can you repeat that?
So,
They have what?
which part? So
The one.
genes contain both introns and exons, right? The mRNA, when it's initially made, contains both introns and exons. And then one of the processing steps is the splicing of it, which is the removal of the introns. Regulatory elements. Yeah, so often these contain regulatory elements. They don't have to. Many intronic sequences have been identified that have no purpose, although perhaps that purpose hasn't been yet discovered.
I think the evolution.
Yeah, and that's why it's thought that a lot of these sequences don't have a purpose, because it's just like an evolutionary remnant. All right, so how these introns and exons get organized, makes this much more complex than we thought it used to be. Here's what I just drew up there. So you get introns and exons in chromosomal DNA, you get transcription, they're still there. And then through this modification, which we call splicing, we get the removal of the introns and the coming back together of the exons. And this can be taken a further step. We're going to talk about it, I think, in this section, which is called alternative splicing. Right? And what can happen is sometimes some exons can be omitted, and you actually get a whole bunch of what are called splice variants for every mRNA. All right, typical human gene size is about 56 kb. So that's the size on the chromosome, right? That's 56,000 base pairs. Right? So the average gene size is pretty big, but a majority of it are exons, right? These larger, a majority of it are introns, these larger pieces of non-coding DNA. So 52 kb of that, on average, is just intronic. Right? A very small fraction of that original genetic sequence is actually coding, actually used by the ribosome to translate into an amino acid sequence. Okay. Introns are really old, right? We know that. They've been around. They're in plants and animals, so that tells us that they're very old. They're very important in regulation and also required to do alternative splicing, which we'll learn about in a minute. But basically, alternative splicing is the production of many splice variants, many variations of a single protein. You can also have what are called nested genes. These are entire genes within genes, right? And these are not extremely common. I think in eukaryotes and humans, it's like less than 1% of genes are actually nested genes. But why would you have this? Well, if you need to absolutely ensure that when you express one gene, another one gets expressed, this is one way to do it. If you take that gene and you actually nest it within that, good figure here. So here you've got your host gene, and you've got the exons and introns kind of dispersed out on one strand of the chromosome. So that strand is going to get transcribed, but nested on its homolog, you have another gene that contains here two exons there's going to be machinery that's going to direct that side to get transcribed as well. So these will get transcribed simultaneously, and you end up producing two proteins at the same time. Okay. So nested genes, when you have one functional gene product located within another functional gene product. Okay, you also have what are called regulatory sequences. These are the sequences that make up promoter regions. Typically, they'll have like DNA binding domains there, regions that proteins can bind to that contain DNA binding domains. These include enhancers, which enhance transcription. They increase the level of transcription. These regulatory elements are ultimately what's required, or what's responsible, rather, for alternative expression of genes, right? So, and defining cells. We'll have a whole chapter on these regulatory sequences. All right, finally, alternative splicing allows multiple protein products to come from one gene. Okay? So through that splicing, you're removing the introns, but through alternative splicing, you can actually omit certain exons in different versions of the gene. Look here, you've got your chromosomal DNA. It's got exons and introns. You transcribe the whole thing. That pre-mRNA has the whole thing. And then during splicing, you remove out the introns. You keep the exons, but oftentimes you'll omit one and it produces different splice variants, right? So here, this one's missing exon 3. This one is missing exon 2 and 4. And so on. This one's missing 2, and that's it. Okay? All these are different splice variants. And I got to tell you, this is what makes, cell biology such a pain. It would be really nice if there's just one gene product per gene, but it's not like that. For every protein, you usually have like 10 or 12 different splice variants that you have to study as well. But this is what provides all of that diversity at the molecular level to do very complex tasks. These different splice variants might have, a job that is related to each other, but they can provide, a different niche functionality to that job. Okay. All right. How are we doing? Great. All right. I'm just going to go a little bit, and I'm about to wrap out here, so I'm getting tired. All right. So this section focuses. All right. You guys can go home. That's it. I think you got enough. All right. So exam 1 is just going to be up to 4.1. We'll put it 4.2 in exam 2. Yes, ma'am.
So we're, just to make sure, the only section we're not doing are from chapters 1 through 4, or 1 through 4.1, it's just 1.3.
I think that, that one section on like microscopy
Yeah,
and stuff,
like
I'm not gonna put anything in there. If I don't go over in class, it's not on the. All right, you guys study hard this week, okay? I want you to do well on this first test, get off to a good start. Have a wonderful week.
Maybe. So.
You shut off this thing.
So when you were describing the little hump with the addition of the
The site-directed mutagenesis.
so what is